Hi everyone, welcome to another class. Here we will discuss about the PCR. What is the full form of PCR? That is Polymerase Chain Reaction. Actually, this is the part of biotechnology, and we know that in biotechnology process there is requirement of cutting and pasting of the DNA segment. Okay, ଆମେ କାଣା କରୁଛୁ? We have to isolate a DNA from a particular organism. ଗୋଟେ particular organism रे देखला ଗୋଟେ organism रे ଗୋଟେ gene ଅଛି, and that gene is, we know that gene is responsible for a particular character, or that gene will responsible for the production of a particular product. If we want that gene, first of all we have to isolate that gene from that particular organism, or isolate that particular organism from a donor organism. Okay, so after isolation of DNA, then we have to put this in a vector DNA, and when the vector DNA and the gene of interest they combine, they will lead to formation of a recombinant DNA, and that recombinant DNA is transferred to a host, and in that host we get the product. Okay, this is the basic features that is taking place in case of your biotechnology process. Okay, so what is the role of PCR in case of biotechnology process? Okay, as I have already told you that this is, suppose this is the organism, and from that organism we have to isolate the DNA. Okay, and that DNA will be combined with the vector. Suppose this is the vector DNA, or that may be plasmid or bacteriophage DNA or any type. And this is the gene of interest, or the DNA segment that is isolated from this organism. Okay, next this vector is transferred to a host, and in the host the gene will express. This is the normal process of biotechnology. We have to cut the DNA from this organism, we have to pass this with the vector, then this vector is transferred to the host. This is basically dealing with the cutting and pasting of the DNA in case of biotechnology process. But when we isolate the DNA, okay, if we have already isolated the DNA, there may be chances of loss. Okay, because we know that DNA is very small. DNA is a very small segment. Okay, as this DNA is very small, and during our work there may be chances that we can lose that. So avoid these kind of things, we have to multiply this DNA into several copies, or we can't lose that DNA segment. So for the multiplication of this DNA, will follow the new technique that is called as what? That is called as polymerase chain reaction, or PCR. Okay, so PCR, that is what? It is dealing with the multiplication of desired DNA. Okay, it is dealing with the multiplication of the desired DNA. Desired DNA means the gene of interest, that is called as desired DNA. Okay, the gene which we want, that is called as desired DNA. Okay, as we know that in case of DNA replication, there is also multiplication of DNA. When DNA undergoes the process of replication, there will be formation of number of copies of DNA. Okay, so first of all, this PCR was discovered by the scientist Kerry Mullis. Okay, he got the Nobel Prize for his discovery. As we know that the DNA undergoes the process of replication, and that will lead to formation of number of copy. So same thing will occur in case of PCR, but here this is in a faster way. Okay, means बहुत कम समय भितर we can get more copies of DNA. Okay, so here also there will be DNA replication. In case of PCR also there is DNA replication, but this is in a faster way. Okay, but this is in a faster way. In very small fraction of time we can able to get more copies of DNA. Okay, that is the polymerase chain reaction. Okay, and this technique is success, or this is possible due to the discovery of the enzyme that is called as Taq polymerase enzyme. Okay, Taq polymerase. Okay, and this Taq polymerase that is isolated from the bacteria that is Thermus aquaticus. This is isolated from the bacteria that is Thermus aquaticus, and this Thermus aquaticus bacteria that is found in the hot spring water. This is found in the hot water springs. Okay, as the Thermus aquaticus bacteria that is heat stable. Okay, this Thermus aquaticus bacteria that is heat stable. As this is heat stable, then the enzyme which will be found in this bacteria, then that will be also what? That will also heat stable. Means in a high temperature, the Taq polymerase can able to work. Okay, बहुत अधिक टेम्परेचर जदि हमें दौछु सप्लाई करूछु, then also Taq polymerase काना करि परबा वर्क करि परबा. Okay, and आकि जितेला हम देखबा mechanism रे there is requirement of this Taq polymerase enzyme, and Taq polymerase enzyme रो discovery ही परिथिला बोली करी PCR get success. Okay, nowadays other enzyme are also used. One of that enzyme that is PFU polymerase. That is PFU polymerase. Okay, what is that PFU? That is Pyrococcus furiosus. Okay? And another polymerase enzyme is also used, that is band polymerase enzyme. Okay, so these are the polymerase enzyme which we were requiring in the time of your PCR. Okay, so the PCR is basically dealing with the repeated DNA replication. Okay, तो जदि हम देखुछु बेसिकली काना होचे इन द PCR repeatedly DNA रो replication हबा. Okay, तो जदि हम mechanism के देखुछु what is actually occur in the PCR or the thermocycler machine. If we see the mechanism, this is occurred in a machine that is thermocycler. Okay, suppose this is the DNA, and this is running from 5' to 3' direction, and this is running from 3' to 5' direction. And if you want this DNA segment, okay, this is the gene of interest. Okay, so if this is the DNA segment, first of all, as we know that DNA replication, during DNA replication, first of all, what is occurred? First of all, both the DNA strand they got separate. Okay, as you know that in natural process there is requirement of the enzyme DNA helicase. Okay, but here we will provide very high temperature, that is about 94 to 96 degrees Celsius. Okay, इनमें क्या करमा DNA के जब 94 to 96 degrees Celsius provide करमा, there will be breaking of the hydrogen bond. Okay, the hydrogen bond will break. Okay, when hydrogen bond will break, there will be separation of the DNA strands. Okay, and there will be separation of the DNA strands. This is 5' 3' and this is 3' 5'. Okay, and this process of separation of the DNA strand during the PCR, that is called as what? That is called as denaturation process. That is called as denaturation process, and this is the first step of the PCR. Okay, in PCR, in the first step there will be denaturation or the separation of the both the strands. Okay, when we will provide very high amount of temperature, that is about 94 to 96 degrees Celsius. Okay, the next step, okay, this is the DNA part we require. Next step, In the next step, as you know that the DNA polymerase is not able to join the fast nucleotides. So that in a natural way, we require a small RNA primer. Okay, that in naturally there is requirement of a small RNA primer, but here we will take a small DNA primer. Okay, or that is also called as what? That is also called as oligonucleotides. That is called as oligonucleotides. And these oligonucleotides, that is the primer that is required here. Okay, as this part here, in case of this DNA, the new strand will form from this direction. Okay, from this direction to this direction. As you know that DNA replication occurs in the 5'3' direction. Okay, so here this will occur in this direction, 5'3' direction. Okay, so here we will require two number of primer, one primer from this and another primer for this. Okay, and this primer are very small strands of nucleotide sequence, DNA nucleotide sequence, that is about 10 to 8 nucleotide sequence, 10 to 18 nucleotide sequence. These oligonucleotides are 10 to 18 small strands of DNA. 10 to 18 nucleotides will be present there. Okay, so after that, the small oligonucleotides or the DNA primer has to be joined with the template strand. This is the template. Okay, this is the template for the new strand, and this is the template for the new strand of this DNA. Okay, both the strand act as template strand for the new strands. Okay, so this primer has to be joined with this template strand. This primer will have to be joined with this template strand. Okay, and for the joining of these oligonucleotides or the primer, there is requirement of very small, as compared to your denaturation process, there is requirement of lesser amount of temperature. That is about what? That is about 40 to 60 degrees Celsius. Okay, and this process of joining or the combination of this RNA primer with the template strand, that process is called as what? That is called as annealing process. This is called as annealing of primer. Sorry. Okay, for the annealing of primer with the template strand, there is requirement of lesser temperature as compared to the as compared to your denaturation process. Okay, so after the annealing process, this DNA primer has to extend. This DNA primer has to increase its length. Okay, and this is possible due to the involvement of the enzyme, that is Taq polymerase enzyme in case of PCR. Okay, now there will be requirement of the DNA polymerase or the Taq polymerase here, and this Taq polymerase will working in this direction for this new primer, and for this primer it will working in this direction. Okay, so after that, next step, that is the extension of primer. There will be extension of primer. Okay, this primer will extend in this direction, this primer will extend in this direction with the help of what? With the help of Taq polymerase enzyme. With the help of Taq polymerase enzyme. Okay, and other raw materials which we will require during this extension of primer, that is your DNTPS. Okay, what is the DNTPS? That is deoxy ribonucleotide triphosphates. Okay, deoxyribonucleotide triphosphates, as well as we also require magnesium ion. Okay, these are the requirement or the raw materials for the extension of primer. As we know that in the for the extension of primer, what will be joining here? There will be joining of the nucleotides. Okay, and these nucleotides, that is from your deoxyribonucleotides, that will be required for the extension of the primers. After that, what will be happening here? This is the diagram of your extension of new primer, and this is running from the 5' to 3' direction, and this is running from the 3' to 5' direction. And suppose this is the primer, and this is running from this direction, and this is the primer, and this is running from this direction. Okay, now 2 copies of DNA will form. Okay, this is one DNA having both the strands. This is another DNA with having 2 strands. Okay, and this is completion of one cycle of the polymerase chain reaction. Okay, again when the temperature will increase, that is about 94 to 96 degrees Celsius, again the denaturation process will occur. Before that, for the extension of primer, again we have to increase the temperature, that is about 72 degrees Celsius. Okay, so for the extension of primer, there will be a requirement of 72 degrees Celsius, and when the 2 strands of DNA are again formed, or the 2 copies of DNA again form, again the same process will occur, same cycle will occur, that is again the denaturation process will occur, again annealing of primer will occur, again the extension of primer will occur. Okay, so this cycle will be repeating. Okay, this cycle will be repeating, and so that there will be formation of billions of copies of DNA in a very small fraction of time. Okay, so after repeated cycle, there will be formation of number of copy. Okay, so if we see a formula, okay, the number of copy formed in DNA replication in the PCR, the number of copy formed in the PCR, that will be 2 to the power n. Here n stands for what? Here n stands for number of cycle. Okay, how much this cycle will occur? How much time this cycle will occur? That will decide the number of copy of DNA will form. In one cycle, there will be formation of 2 DNA. That means 2 to the power 1. One cycle 1. 2 cycle 2 to the power 2. That means in the second cycle there will be formation of 2 to the power 2. That is equal to 4. Okay, in this way, suppose 3 cycles occur, 2 to the power 3 is equal to 8 number of DNA will form. Okay, so this cycle will repeating, that will lead to formation of number of DNA copy in a very small fraction of time. Okay, and nowadays this PCR technique is very common, so that now some people call this as people choice reaction. Okay, as this is now very common. Okay, and this is the all about your PCR. So before we close this section, what are the requirements of your PCR? One is the DNA, or the part of the DNA segment. Okay, so DNA segment for its amplification. Amplification means increase its number. Okay, then another requirements is that DNTPS, deoxyribonucleotides triphosphates. Then there will be requirement of your tech polymerase. Okay, and there is also thermal cycle. Okay, thermal cycle means increase and decrease of the temperature. For the denaturation process, there will be requirement of 94 to 96 degrees Celsius. For the annealing of DNA primer, there will be requirement of 40 to 60 degrees Celsius. And for the extension of primer, there will be requirement of 72 degrees Celsius. So this is the temperature is going on increasing and decreasing during the time of your PCR. Okay, so this is all about your PCR. Okay, and why we should need this PCR, what is the application of PCR, we will discuss in the upcoming process.